Good morning, good afternoon, good evening to everyone. I'm Jennifer Smilowitz, the Director of Scientific and Strategic Development for the IMGC, which is hosting today's webinar. Thank you all for being here today. We have 238 registrants from so many parts of the world, from the United States to countries in Europe, but New Zealand, Israel, Indonesia, India, Chile, Mexico, China, Pakistan, Sri Lanka, and South Africa. Thank you all for being here. It's exciting to launch our first virtual webinar series called ROS for Roll Up One's Sleeves. The goal of this webinar series is to focus on one translational issue in milk science and lactation, to identify key hurdles to scientific development and recruit innovative solutions from a broad scientific audience. Moving forward, each year, the IMGC ROS workshop will focus on a translational issue that's new in milk science and lactation. We have identified key experts in the field and asked them to share insight from their experience, to think about what they wish they knew 10 years ago or more that they could use today to improve some kind of method or outcome. And so we hope that you come away with a better understanding or learn something new on the topics discussed today. This webinar is formatted to showcase 5 back-to-back presentations, followed by a 5-minute break, and then a 1-hour Q&A and an open panel discussion with the speakers and the IMGC Scientific Advisory Committee members. Please submit your questions in the Q&A box throughout the entire webinar. I will compile them as you submit them and then share them during the Q&A session. Also, please complete a 5-minute online exit survey at the end of this webinar. This is so you can share your experience, you can tell us what you liked, what you didn't like, and then you can share with us what you'd like to see covered next year. A link to the survey will be shared in the chat box, will be shared in the Q&A box and the chat box, as well as a QR code in my closing remarks. The webinar is being recorded, and so it'll be available on the IMGC website in the next couple of weeks. So before we start the webinar, I'd love to share with you a little bit about the IMGC. So the IMGC is a scientific society, and it was founded in 2004 by Bruce German at UC Davis and colleagues with the support of CDRF, the California Dairy Research Foundation, which currently co-manages the consortium. Now, the original goal, the reason why people got together in Napa in 2004 was to sequence the bovine lactation genome, hence the name milk genomics. Now, this was accomplished in 2009, which was published in 2 papers in 2009. And the group continued to meet annually because there was just still so much to discover about milk and lactation. Currently, IMGC includes hundreds of scientists and innovators worldwide from different sectors, including academia, industry, policy, government, NGOs, etc. The overarching goal of the IMGC is to discover and share the functions and impact of lactation and milk for human health. The word genomics in the consortium's name stuck because all functions in lactation result from mammalian genes. So how do we advance the fields of milk science and lactation to improve human health? Well, it's been, for more than a decade, we've leveraged several platforms that offer different mechanisms to make an impact and advance the field. Let's review them. So our annual symposium is our flagship conference hosted in the fall every other year at UC Davis, and then somewhere else in the world in alternate years. And this year we're hosting our 19th annual symposium at UC Davis. This 2.5-day event features cutting-edge scientific research that accelerates scientific innovation, advances and identifies actionable opportunities, and attracts a diversity of disciplines and fields from chemistry, microbiology, immunology, engineering, bioinformatics, anthropology, and even other stakeholders are involved, such as farmers and clinicians and policymakers. Participation in IMGC symposia has shown to statistically increase collaboration, increase the number of publications and number of publications in high impact factor journals, and recruit new scientists into the field. And this has been shown in Kwok et al. In PLOS One. So juxtaposed to the symposium is our awards program, which is instrumental in highlighting scientists across different career timelines, from students to tenured scientists, for innovative, cutting edge, and impactful research. This program also encourages ongoing engagement and builds a strong scientific community. Our third platform is our translational arm for a broad scientific audience in the form of a bi-monthly publication called Splash Milk Science Update. Splash is written by professional science writers who translate the latest scientific discoveries in lactation, milk, and dairy science from primary scientific peer-reviewed literature. We publish 4 articles per issue, which resulted in 432 archived articles that you can search on our website, some of which have identified hundreds of potential biotherapeutics and novel applications for human health. We have more than 200,000 unique page views online, with the majority of readers coming from social media. We have readers from 5 continents and in every major city in the world. Now, welcome to our newest and 4th platform, the Roll Up One Sleeves workshop, which is designed to focus on translational issues in lactation and milk, but for a scientific audience, hence you. The workshop was created to identify key hurdles to scientific development. We're recruiting innovative solutions using a broad range of scientific principles from experts from all over the world. The learning environment is focused and detailed as on one specific topic, as opposed to covering a breadth of topics as intended in the symposium. You should come away with a better understanding of bringing discoveries of science to practice as concepts or products for human health. And this year's theme is scalability, analytics, and navigating regulatory, which is a focus on milk peptides and proteins. Please join world experts in milk science as they share with you their insight and experience in dealing with the challenges and finding solutions for overcoming those challenges in scaling up analytics and navigating regulatory. This topic is extremely timely given the recent infant formula crisis in the United States. And several of the speakers will address ways to approach safety with a new lens. Let's get started. It is my pleasure to introduce the co-moderator of today's webinar and member of the IMGC Scientific Advisory Committee, Professor Casper Hettinga from Wageningen University and Research in the Netherlands, who has also been instrumental in co-developing the webinar's program along with the IMGC Workshop Committee. Casper, please take it away.
Thank you, Jennifer, for the kind introduction. And I'm really happy that we now have this first annual workshop. And I will not say much more because we will get started immediately with the contents. The only thing I want to remind you is what Jennifer already said. If you have any questions to the speakers, please write them in the Q&A, because in the final hour of today's workshop, we will have a panel discussion and we will use the questions from the Q&A as part of this. But now I want to introduce to you Professor Daniel Tomé. Daniel Tomé belongs to AgroParisTech and has served many times on different EFSA panels. Today he will give a lecture entitled Navigating Regulatory for Dietary Bioactive Peptides and Their Health Claims. Daniel, please take it away.
Okay. Hi, everybody. Thank you for the invitation to give this talk. So my talk refers to the regulatory aspect for dietary bioactive peptide and their relation with the possibility to apply for health claims. So this is the disclosure as usual. And so it has been now established for a relatively long time that some food protein and peptide, which are encrypted in the sequence of this food protein, are potentially bioactive. There is a lot of bioactivity that has been identified, for instance, the opiate peptides, angiotensin converting enzyme inhibitor, immunomodulatory peptide, and so on. I will come back, and Over will come back also. These sequences have been actively reviewed, particularly for milk protein and peptide, but also there are studies related to other sources of protein like animal, plant, sea, and microbial products. And so the question of this talk is to evaluate whether this bioactivity can be used and can meet the criteria for health label claim for conventional food, dietary supplement, and infant formula. This is a very important question, particularly for research and innovation in this area, because if to promote the innovation in this area, there is a need to be able to say something after about the properties and the activity of this compound. And then we know that then it is related to health claim and the possibility to have health claim for food. We know now we more and more strongly regulated by national and local authority. And then so there is a need to clarify this question. So what can food protein derived peptide meet the criteria for health label claims, and what are these label claims which are subjected to evaluation and authorization by different authority? So health label claims are defined and authorized by expert authorities and expert committee for food and health. Two important agencies are the European Food Safety Authority in Europe and the Food and Drug Administration in North America. There are other authorities which are involved in the health claim, but for instance, in Japan and in other countries. But we know that these two agencies are very important and probably the leading agency in this area. So what are the health label claims? So they relate to food components and their possible effect on nutritional intake, including infant formula, improvement of physiological function in the body, or even strongly, the prevention of pathology. And in this case, there is a need to have a clear regulation to clearly protect the consumer. So for the regulatory aspect and the document related to health label claims for the food, even conventional food, dietary supplement, and infant formula, so there is relatively now clear definition, particularly by FDA and EFSA, of the different category of claims. So the more simple one, so are what we call the nutrient content. So they relate to generic nutrition claims that describe the property of nutrient with reference to an abundance, and then it explains what can be said about each nutrient. A bit stronger are the structure function claim. They relate to development, maintenance, or improvement of a function. Their authorization is granted on a case-by-case basis, and they lead either to generic function, meaning that it can be used by any producer of the corresponding food, or to exclusive property of the applicant when there is a requirement to protect some of the food developed by one specific company. And so the stronger possibility is to apply for disease-related claims that describe the effect of a product and compound on the reduction of the risk of disease. It is a very strong claim, so it has to be protected. It also refers usually to children development and health, like the property associated to infant formula. More usually, their authorization is granted on a case-by-case basis. There is an additional category particularly described by FDA, which are the qualified health claim for claim not granted by FDA. In this case, at least in America, in US, the applicant is able to say something about this claim, but he has to indicate that the claim was rejected by FDA. This does not exist in Europe. There is no label possibility when a claim is rejected by EFSA. It's not allowed to say anything in this case. So this is the general picture of the claim. So what is important is to clearly understand the condition which had to be met to obtain a health claim, and there is three main conditions. First, food bearing the claim must be precisely described and reproducible. Secondly, the targeted function or effect must represent a benefit for the consumer and must be measurable. This is very important. And thirdly, the claim effect must be demonstrated in humans on the target population. So here the first one is very important because the consumer has to know what constitutes the food bearing the claim, and to be sure that the composition of this food and the properties of this food can be reproduced. This is a guarantee for the consumer that what is claimed can be really obtained with the food. This is probably the more important condition because we know that food can modify many functions. In some cases, there is no clear consequence for the consumer. So in this case, when there is a modification of a function, it should be a benefit for the consumer, and to be sure that it can be described, it has to be measurable. So there is a need of biomarker of the effect and so on. Something has to be measured. And so, important also aspect, it has to be demonstrated on the human population and particularly on the target population related to the health claim. So in the condition, studies are carried out in conditions representative of those for which the claim is intended. So as I say, there should be sufficient definition of a food constituent. It should be reproducible when the claim is made. And so the condition under which the human studies are performed should relate to quantity and patterns of consumption of the food for which of the claim is made. It should be studied in conditions which are completely different to the condition for which the claim is made when the food is consumed by the population and by the consumer. So this is an important part of the reflection. As I say, the target effect should relate to a benefit for the consumer. What does it mean? For function claim, a beneficial effect may relate to development, maintenance, or improvement of a function. In this case, so it could be a benefit. And in the more strong claim related to reduction of disease risk, beneficial means refers to the reduction of a measurable risk factor as independent predictor for the development of a human disease. So there is clearly a need to identify a function or a reduction of disease that will be a benefit, and that there is a measurable risk factor of this effect. And so in this case, it should be performed through experimentation with leading to a significant scientific agreement on the effect. So it is the case for both authorities. So when the FDA evaluates a health claim, the agency considers the totality of the publicly available scientific evidence, and this includes well-designed study with generally recognized scientific procedure and principle. That means some study can be rejected because they do not agree with the usual way to perform such study. And then if there is a significant scientific agreement, then the agency will authorize a health claim for the substance, Quite the same in Europe with the European authority in the scientific substantiation for claim in Europe. Nutrition and health claims are based on and substantiated by generally accepted scientific evidence. Always in both cases, there is this problem of scientific evidence recognized by the scientific community. Human studies are central and should use appropriate, generally accepted outcome measures of the claim effect. So when we go to the peptides, so we can find now many bioactivity associated to milk protein derived peptide, for instance. I did an exhaustive list. We know that there are many bioactivity that has been found in casein, beta, alpha-S1, kappa-casein, and also in soluble protein, alpha-lactalbumin, beta-lactoglobulin, and lactoferrin. So in many cases, you know, there are many peptides with opioid agonist activity and with angiotensin converting enzyme inhibitor activity. And you see they are found in many of these proteins. Overactivity related to immunomodulation, mineral absorption, antioxidative effect, anti-hypertensive effect, antimicrobial growth factor, and so on. Over many activity, but this activity are not per se a benefit. So we have to decide which peptide, which bioactivity is carried by the peptide, and then to associate this after the consumption of the peptide to a function, to an effect in the body, and to be sure this effect is a benefit. And so this is the question that has been addressed, has to be addressed by any applicant for health claim to choose the functional effect, at least not also exhaustive. This can be antimicrobial, antioxidative, immunomodulatory effect, infection resistant, gut function, nutrient carrier, vasoactive activity, anti-hypertensive, body weight control, infant growth. I list it because this effect has been proposed in different dossiers of applicants. And then the first question to address, and the second one is, can you associate to this type of effect a benefit? As you can see, it can be discussed. For some, for infant growth is clearly a benefit. Body weight control is very effect. It's a benefit clearly. Gut function are usually recognized as benefit. For other, it's more controversial, and the applicant has to go in more detail to the effect. And for the measurement, also we know the biology is a very complex domain, and so we have to discuss. For example, antioxidative is a good example. In many cases, it's very difficult to define its benefit, what has to be measured, and so on. So this is the beginning of the building of a dossier related to the application for a health claim. So some example of claims and benefit and measurability. So when claims for claim on bowel function and gastrointestinal discomfort, for instance, is there benefit? First rule, claims related to bowel function are usually beneficial physiological effect, provided they do not result in diarrhea. So this is the first condition. Then, if there is no effect, suspicion of effect on diarrhea, different aspects, reduce transit time, gastrointestinal discomfort, more frequent bowel movement, increased fecal burden, shorter tools are beneficial physiological effect. They can be measured directly by gastroenterology approach, or also even very often it can be performed using validated questionnaires, some of which are very good and can be used and can be accepted by authority to accept claims when it clearly demonstrates an effect on this function. Or example for claim related to cardiovascular health, we know that there are validated biomarkers, whereas others are not validated and should not be used. On the validated biomarkers, some are very traditional now, LDL and HDL cholesterol, triglyceride, blood pressure, specific endothelial function, platelet aggregation, normal homocysteine metabolism are recognized biomarker of cardiovascular health risk, recognized by the clinician community. And so they can be used. The applicant just have to show that there is an effect on one of these markers. In contrast, for instance, endothelial function per se is not sufficiently defined according to the scientific community and cannot be used alone to demonstrate an effect on cardiovascular health. And last example, very often there is claim application for effect of gastrointestinal microbiota. Is there benefit? It's probably more controversial and difficult to demonstrate, although it's a very interesting area. Huh? Based on current scientific knowledge, it's not possible to precisely define a normal microbiota. So this is the main difficulty. It is known that even increasing the number of some specific group of microorganisms, like lactobacilli and bifidobacteria, is not clearly in itself a beneficial effect. It should be associated to something else. Is it measurable? Yes, the composition of the microbiota now is relatively, even in some cases, quite expensive, but it's not difficult to have the composition of microbiota. And we know it has been clearly shown that the gastrointestinal microbiota can be altered by food constituents. So the beginning of the story can be constructed, but per se, because there is no clear reference on the normal microbiota, it should not be used alone. And for function claim related to change in the microbiota, in the gastrointestinal microbiota, this change has to be associated to other beneficial physiological or clinical outcome directly measured on gastrointestinal function, cardiovascular health, feeding behavior, and so on. And so it should be associated to other measurements. Okay, so as a conclusion, some bioactive peptide potentially meet the criteria required by regulatory authority for obtaining a health claim. It's clear there is many bioactivity. We know that the bioactivity are associated to body function, to disease risk, and so on. And so it can be imagined that in the future, we could develop a new strategy to use this component to modulate health. But in this case, as I say, there is clearly a need to meet the criteria required to obtain health claim, either function claim or decrease in the risk of disease. An important criteria is to target function or effect that represent a clear benefit for the consumer and which are measurable. If there is no benefit for the consumer, even if a function is modified, if it's not a benefit, it's not possible to have a claim. And to demonstrate this, there is a need to measure something. Currently, a major limitation is the lack of convincing clinical studies conducted according to validated clinical practice. We know that it's very complex to conduct clinical studies, particularly in nutrition. Many parameters are involved. It's very complex. There is a lot of variability among people, polymorphism in human population. This is a very difficult area, but to obtain claims, there is a need to demonstrate the effect directly in human. Comment for infant formula, their authorization is also granted on a case-by-case basis, like the other claims, how to go faster in a context of shortage. This is a small joke at the end. Thank you.
Wonderful, Daniel. Thanks so much for kicking off this workshop. You have given a very nice introduction of how to look at novel ingredients in infant formula and in other foods from a regulatory point of view. I can imagine that there are people who have some questions about what they have just heard. So I want to remind everyone again, please write your questions in the Q&A and we will deal with all the questions at the end of the workshop in the final hour. So now I would like to introduce our next speaker, which is Dr. David Dallas. He's an assistant professor in nutrition at Oregon State University and also a member of the IMGC Scientific Advisory Committee. He will give a lecture entitled Overcoming Challenges in Analytics of Milk Proteins and Peptides and their Digestome. Please, David, take it away.
Okay, thank you, Casper. Thanks to the IMGC for inviting me to share my perspectives on the analytical challenges and solutions for getting effective novel protein and peptide additions translated into humans and on the market. So I have no personal financial interest to declare. So milk, as many of you know, is the product of more than 250 million years of evolution to provide the complete nourishment system for the mammalian infant. As each component in milk comes at a cost to the mother, we know that each component must be beneficial for the infant. In terms of protein nourishment, milk includes some proteins that are easily digestible to provide a balanced array of essential and non-essential amino acids that the infant can use for protein synthesis. Yet, proteomics research demonstrates that milk contains hundreds to thousands of unique proteins. These findings suggest that milk provides more than a simple amino acid nourishment system. Indeed, many milk proteins have been annotated with function in their intact forms, such as antimicrobial, anti-adhesive, guiding infant development, immunomodulation, and calcium delivery. Many of these proteins may serve as precursors for hidden bioactive peptides that are released upon interaction with proteases in the infant gut. These functional peptides have known activities including killing bacteria, preventing pathogen binding to intestinal cells, feeding beneficial bacteria, modulating the infant immune system, increasing calcium uptake, stimulating the production of mucin, which is the coating that coats the intestine, and binding to opioid receptors in the gut, which could alter the rate at which food moves along the digestive tract. So milk contains an amazing array of bioactive proteins and peptides. And the field, including many in this audience, have worked incredibly hard to identify the molecular composition and functions of proteins and peptides in human milk. As we know that all the proteins and peptides present in human milk are likely beneficial to the infant and surely safe, most of these components could be translated to improving human nutrition, both for infants and for older people. Yet, most of the known bioactive proteins and peptides discovered in milk have not yet been translated into functional nutritional products. And this is an enormous opportunity gap. Fortunately, we now have the tool sets to greatly accelerate the pace at which knowledge about human milk proteins and peptides can be translated into real health benefits for people. Researchers in industry and academia continue to refine methods for scaled up extraction of key bioactives from bovine milk. For example, what you will see from the talks of Roland van Aerten at Friesland Campina for lactoferrin, and from Anne Stout-Kvistgaard and Peter Langborn-West at Arla for osteopontin. Increasingly, the tools are available for the production of milk bioactive proteins using recombinant expression systems, as Laura Katz from Helena will discuss. One key challenge in this space is determining which molecules in human milk should be early targets for supplementation to formula. Formula is an obvious and essential area for translation of human milk bioactives to improving health outcomes. We know that formula in its current form provides poorer protection against negative health outcomes than human milk, including higher rates of necrotizing enterocolitis in preterm infants, higher rates of allergies, and potentially poorer neurodevelopment. Yet, there are a large number of infants who do need formula. The lower effectiveness of formula compared with human milk derives from the differences in their molecular composition. So increasing the similarity of the molecules in formula to that of human milk is a key goal to solve for. How can we identify which bioactive proteins and peptides are good early targets for supplementation to infant feeding? We can't just base this choice on what we know about bioactivities of proteins present in milk itself. Once milk is ingested by the infant, its component proteins interact with an array of digestive proteases and pH changes, leading many proteins to be degraded, including many to free amino acids to be used for infant protein synthesis. To be biologically relevant in an active form within an infant, a bioactive protein would need to survive the onslaught of digestive elements to stay intact to its site of action within the gut or systemically. To identify candidate proteins for supplementation for infant formula, we need to determine the extent to which they survive intact within the infant gut and can be biologically relevant. Similarly, proteins may serve as carriers for bioactive peptides that are released upon exposure to proteases during digestion. As most known bioactive peptides were discovered from dairy products or after in vitro digestions, we know very little about which survive to or are released at their predicted sites of action, and thus whether they are biologically relevant. To identify which peptides or protein precursors are important to supplement to infant formula to exert bioactive peptide function, we need to determine their survival and presence within the infant gut. A key solution to this challenge is the ability to determine which proteins survive and which peptides are released from digestive samples taken from human infants. We have partnered with collaborators at neonatal intensive care units in hospitals to collect gastric fluid from indwelling gastric tubes. And later, we've been able to identify the possibility of extracting intestinal fluids using nasointestinal tubes. So we're able to collect gastric and intestinal digest from human milk. To analyze these samples, we developed special methods to extract the parts we are interested in, peptides or proteins, out of the mixture. For intact proteins, we perform trypsin digestion for bottom-up proteomics. We inject these extracts into a liquid chromatography instrument, which helps separate these molecules, and analyze them via mass spectrometry, which allows determination of their molecular composition based on highly accurate mass. We have worked to optimize each of these steps, including compound extraction, liquid chromatography, and tandem mass spectrometry methods and data analysis. For peptides, to help interpret our data, we scoured the literature on bioactive peptides, including from a variety of different sources like human, cow, goat, and sheep, and created a comprehensive database of over 1,000 known bioactive peptides. And we implemented an algorithm that can be used to search large data sets of peptides against these known bioactive peptides to search for homology. Our group has analyzed infant gastric, intestinal, and stool samples from preterm infants and found a large array of peptides highly homologous with known bioactive peptides with antimicrobial, calcium binding, antihypertensive, immunomodulatory, and opioid activity. These bioactive peptides found represent potentially biologically relevant targets for supplementation. In addition to those with known function, we found thousands of peptides without any known function. And these uncharacterized peptides need to be assessed for functions via testing interactions with various gut relevant cells, like epithelial cells, immune cells, and gut bacteria. So this can lead to novel peptides that can be target for supplementation. To guide targets for novel protein and peptide supplementation to formula in the future, we need much more analysis of the extent to which proteins survive and to which peptides are released using real infant samples. If possible, followed up with work to examine their site-specific bioactivity. And alternative models to this can include simulated digests and animal models like rodents and piglets. However, the complexity of the human gut makes translating findings from in vitro and animal models to understanding milk digestion in infants quite difficult. A second challenge, once we define which proteins and peptides are valuable as supplements based on this information about survival, the produced replacement compounds, either extracted from bovine milk or produced recombinantly need to be tested to ensure they have a similar survival to the extent of survival for human milk. So how can we do this? Optimally, it will be via in vivo human testing in infants with sample collection from the stomach and intestine, as well as stool, followed by mass spectrometry, testing functions, measuring proteins via ELISA, for example. One example of this, we've been able to test the digestion of a recombinant protein called palivizumab, an antibody to respiratory syncytial virus in preterm infants, and compare that with the survival of naturally occurring human milk antibodies. The results were very interesting, though the sequence of the constant region of the antibody were identical between the recombinant form and the natural form. We found that human milk antibodies survive mostly intact throughout stomach and intestine of the infant, whereas the recombinant form was mostly degraded. So this difference in survival could be due to other differences in the protein's folding or post-translational modification, like glycosylation. And this vignette demonstrates how necessary it is to ensure that the survival of the supplemental proteins matches that of human milk, or we could end up feeding expensive and ineffective supplements to infants. So this can really help guide further development to optimally stabilize novel proteins against digestive enzymes and other conditions. When it's not possible to obtain approval for direct feeding of a protein, we can use techniques like ex vivo incubation in collected infant digestive samples to provide an approximation of in vivo digestion. For example, we did this with the recombinant protein palivizumab and naturally occurring human milk immunoglobulins by incubating them in infant digesta. A lower form of evidence, again, could be using simulated gastrointestinal digesta to mimic that of infants. However, of course, the complexity of human digestion is much greater than what we can produce in an in vitro or an animal model. And in the future, novel ingredients for infant formula should be assessed to ensure that their digestive profile provides similar bioactive compounds as human milk. And for proteins intended to provide bioactive peptides, the release and survival of these peptides should be compared between human milk and the proposed supplement. So a third challenge is beyond infants. There's an enormous opportunity to translate milk's bioactive potential to enhance non-infant health, so adolescents and adults. With non-infants, there's also a need to validate the potential benefits of novel milk proteins and peptides within adults, whether produced in isolation from bovine milk or produced recombinantly. Though protein and peptide products may have function in their initial form, whether they're biologically relevant will depend on the extent to which they survive digestion in the consumer. So to address this issue, we can assess the digestion of specific novel proteins and peptides in the gut of consumers. So like in infants, we've been able to fit adults with nasojejunal tubes and collect samples from their upper small intestine. And you can see I'm one of the subjects in this study. So far, we've fed subjects with whey protein isolate from bovine milk and purified bovine milk glycomacropeptide, which is a large fragment of kappa casein that's released typically during cheese making and ends up in whey. And in this case, we were assessing glycomacropeptide digestion because various studies indicate that in its intact form, this peptide exerts an array of bioactivities, including antimicrobial and immunomodulatory, mostly as described from in vitro assays. But we wanted to understand whether GMP, that's glycomacropeptide, has potential to exert these functions in the gut in its intact form. So we were able to extract the peptides from these samples via a variety of different approaches, analyze them by LC-MS, and performed glycopeptidomic analysis using a program called Bionic. And in these studies, we have demonstrated that actually, surprisingly, little to no glycomacropeptide actually survives intact to the adult jejunum. So this finding indicates that previous data on the activity of this glycomacropeptide isn't actually likely relevant to human nutrition. However, we found that GMP glycomacropeptide is digested into an array of peptide fragments. And some of these fragments were highly homologous to known bioactive peptides with functions like antimicrobial and antihypertensive. So it's possible that glycomacropeptide could exert an effect in the adult human gut via its released bioactive peptides. And more work is needed to examine the functional relevance of these released peptides and glycopeptides. So in the future, as this example indicates, we need to assess the digestive profile of each novel protein and peptide product for consumers in the relevant consumer population to determine whether they have biological potential. And then this work can guide follow-on work to optimize the survival of desired bioactive components and to test the specific functions that those components have. So in conclusion, we have a fantastic opportunity to accelerate the translation of research on bioactive proteins and peptides to foods and therapeutics for infants, adolescents, and adults. To guide selection of proteins and peptides for supplementation that will be maximally biologically relevant, future work should examine human infant digesta and adult digesta as well, with advanced analytics to determine the specific forms of milk proteins and peptides present at specific sites within the gut. And then follow-on research should examine their site-specific bioactivities. To ensure that novel proteins and peptide ingredients for infant formula or adults that have the same bioactive potential as their human milk counterparts, they need to be assessed to ensure their digestive profile is similar to that of naturally occurring compounds in human milk. For adults, novel bovine milk proteins and peptides or recombinant forms should be assessed for digestion to evaluate their potential to exert bioactivity within the body. These steps are essential to ensure that we extract the most value from milk's incredible model for human nutrition. Okay, thank you.
Wonderful, David. Thanks for this presentation. After first hearing from Daniel about the regulatory framework and that you need to prove benefits, it's nice to see your approach to actually testing these benefits. So thanks for your presentation. And as already mentioned before, but I will mention it again, if you have any questions to any of the speakers, please type them in the Q&A and we will compile them for the final hour. But now I would like to introduce to you the next speaker, which is Laura Katz. She's a founder of Helina, which aims at making recombinant human milk components. She will give a lecture entitled Building a Framework to Evaluate the Safety of Recombinant Proteins. Laura, please take it away.
Thank you so much. And thanks everybody for joining me today. Really excited to be here with IMGC and to be speaking to all of you today about building a framework to evaluate the safety of recombinant milk proteins and really what we're doing at Helina to address this. Very excited to be speaking after Dr. Dallas, although I don't think I could top that. His work is so critically important, as is everybody's here today. So happy to be part of the conversation. So I think it's clear to all of us here, given everything we've learned so far today, that bioactive proteins prove that food can be more than just nutritive. These proteins found in bovine milk and human milk and really in mammalian milks can help to enhance or aid in nutrient absorption, modulate the immune system, defend against pathogens and infection. And these are all quite desirable properties, especially when we think about infant nutrition. So at Helena, what we do is we are recreating the proteins found in human milk, and we are really focused on bioactive proteins, proteins that help to provide these types of properties to infants and bring this to consumers. And that really has not been done before on a commercial level, although there's been so much research around this. So infant formula is, of course, one application, and it's something that, you know, I'm sure we could all agree on is a very obvious first step in taking recombinant bioactive proteins, or, you know, human milk proteins, and putting them into a consumer product. But really, as Dr. Dallas just spoke about, there's other applications beyond just infants. And we can think about using these proteins as food ingredients in other applications like functional food products. We think about nutritional beverages for people at various different ages, dietary supplements. So there's a really big scope of what it means to take bioactive milk proteins and put them into food. So there's huge scope here. And this is exactly what Helena was built upon, is that we can find these proteins in human milk and in bovine milk and in other milks. And we know that these proteins have these exceptional properties, but taking or fractionating proteins, bioactive proteins out of bovine milk is, while critical, very unsustainable. There's a lot of milk that we need to actually take. And as more and more consumers are moving away from cow's milk or are looking for sustainable alternatives, it becomes really difficult to discuss or sell to a consumer. We're taking, you know, this very small percentage of a protein out of cow's milk. And it's also quite expensive. So alternative methods to produce these proteins will become critical in being able to bring bioactive proteins to consumer products, so we can be bringing the properties of these proteins to consumer products and also making them accessible. Making something that is more accessible from a price point perspective is going to be important as part of coming to a solution to how we can get bioactive proteins into food. So there are a handful of methods that we could use. I'm sure there's probably at this point more than this, but microbial fermentation via a host cell, so training or engineering yeast or bacteria or fungus to make protein, to make these bioactive proteins, using mammalian cell culture and or transgenic crop and animal to create or produce these proteins as an alternative to fractionating or purifying these proteins out of cow's milk. Of course, we're not going to be purifying these proteins out of human milk anytime soon on a mass level or on a consumer level. So these are really great alternative methods to produce these proteins. And now is the perfect time for innovation within this category, going and looking towards these alternative methods to create bioactive proteins on an industry level. We can create a very targeted approach to achieving the native structure. So as we've heard about, and as you know, I think there was a question about post-translational modifications, particularly using organisms like yeast. We can actually target post-translational modifications and other unique characteristics of a protein to achieve the structural integrity of the native protein, which is going to be critical for the success of creating a recombinant protein, is making sure that we do target the amino acid sequence, the post-translational modification, the folding, all of the things that come together to make that protein what it natively is. There's high market value for bioactives, and this may justify the cost of R&D and scale up and all the work that goes into the innovation behind getting these products to market. Bioactive proteins, I mean, they're bioactive, so they're doing more than just providing nutrition to us. And the consumer, particularly within the infant formula category, understand the value and therefore would likely be paying or be willing to pay a higher price than, you know, just a standard commodity priced protein, because we're bringing additional value to the consumer's health. And then the alternative methods that we see, particularly using cell culture or precision fermentation via fungus or yeast or bacteria, is reduced reliance on animal agriculture. As we try, as you know, food industry, to move away from excessive reliance on animal agriculture, these alternative methods become even more relevant and important to being able to bring the same types of benefits that we find from animal-derived products to consumers in a more sustainable way. We think at Helena, and this is where, you know, we're probably a little bit biased because it's the technology we focus on, but precision fermentation is the best approach to scaling the production of bioactive proteins. And this is because the food industry has been using fermentation for years, for a long time, to make all different kinds of food ingredients. And so we already have infrastructure. One of the big hurdles, and with plenty of hurdles along the way to recreate bioactive proteins recombinantly, is how am I going to scale this? We need to be able to take what is, or what could be, a science project to, you know, turn it into something that becomes a business, and that becomes scalable and something that we can actually grow to feed people. And if we're talking about feeding infants, I mean, they eat a lot of formula or they drink a lot of milk. So we need to be able to produce a lot of this protein, or of any bioactive protein that we plan to scale for commercial applications. And so with precision fermentation and using fermentation as a tool, there are manufacturing facilities that exist to do just this across food and drug, that have the tank capacity and just overall capacity for the industry to be bringing in these types of technologies and manufacturing at scale. There are many microorganisms used to express proteins that are already recognized as safe and currently used by other companies within the food industry to make other types of proteins. Particularly, we've seen over the last few years, a handful of companies take new proteins through the GRAS program via the FDA, and the microorganisms that they're using to express these proteins are already recognized as safe, which is particularly important when we think about having to then also understand and demonstrate safety for the protein itself, to start with a expression platform that is safe. And fermentation-made proteins are already in our food supply. As I just mentioned with GRAS, there's plenty of fermentation-made proteins that exist. And so taking this, you know, really taking a technology that's currently really being leveraged to make proteins for their sensory profiles and changing our lens on what it means to use this technology for making proteins with bioactive properties is a really exciting opportunity to really use this technology for good. But then there are, of course, with any opportunity comes challenges, particularly relating to the downstream purification and scaling. The challenges that we see in the industry, I would say, come down to time and cost. So to, come in and engineer your host cell to achieve all the post-translational modifications that you're looking to accomplish consistently, and to, really target any specific characteristic of your protein. That takes time, that takes cost. And especially for new innovators coming into the space, like companies like Helena, where we rely on venture capital funding, we need to be very sensitive about what it means to innovate and be very targeted with our R&D dollars, because it can add up quite quickly. Producing enough volume to achieve an accessible cost, something that frankly we face, that we're working through, and I know plenty of companies face, is as you engineer a host cell, the efficiency of that cell to actually produce your protein of interest can become modified as you really work to engineer the cell. The cell can become less and less healthy. And so you may be producing the right protein, but not necessarily a high volume of that protein. So you need to figure out how to train your cells to produce enough protein to ensure that the process becomes cost effective. And then lastly, downstream purification is, I would say, for us and for many companies in the industry, making recombinant bovine proteins, we know to be one of the biggest challenges. And that's because purification requires a lot of development in large volumes. It's hard to mimic, a manufacturing setting in a little lab. And there are question marks around the different unit operations you would need to bring in to achieve the purity and the recovery that you hope for your protein at scale without having to invest. And so to invest in new unit operations to purify each individual protein differently, because they might all require something different, can become quite timely and expensive, and a hurdle for development. So all of these challenges that we see, I would say, are certainly solvable. They require, big teams of people like we have here at Helena, working to kind of get through the needs of the purification, or the needs of the scale up, or the needs of the specific protein to achieve an ingredient profile that is desirable for the business. The thing that we as a scientific community need to align on in order to not just be able to make and scale and manufacture these proteins, but to get them into the food supply, is to align on a framework to evaluate the safety of these ingredients. And there's really kind of two big categories that we see as areas that we need to address. The first is the structural identity of the protein. So this comes with the amino acid sequence, the post-translational modifications, and the tertiary structure, or the folding, and what that looks like of the protein. I actually think this is quite straightforward, because these are the kinds of things as innovators you should be consistently reviewing in the R&D process of making your protein in a lab and as you scale, to ensure that you're achieving a profile that you believe is the best for what your expected outcome, or what your hopeful outcome is. When it comes to, sorry if you hear the New York City background here, when it comes to dose and exposure, we still have question marks on how much is safe to eat. And if we look at things like bioactive proteins, particularly some of the examples that we've seen recently that are GRAS, or are deemed generally recognized as safe, the dose, or the amount that we are permitted to include in things like infant formula of proteins like bovine lactoferrin is significantly less than the quantity that we would see in human milk. So it's how do we arrive at those numbers, and then how do we assess things like toxicity, allergenicity, immunogenicity, and absorption, distribution, metabolism, and excretion of these proteins in a way that is accessible to the industry. So all of these are things that we need to be able to think about and build a framework around so that we can be ensuring that from every single perspective, what we're making is safe and doesn't have any cause for concern when it comes to any of the things that we've mentioned here. So the framework to assess safety must be reliable and affordable for innovators. I would say this is probably the crux of the challenge, is if we want to take all of this great science that everybody here is working on and translate that into something that will have significant impact on consumers, and particularly infants who right now, you know, as we're seeing right now in the industry, we're really dire for innovation and new products and options for parents. We need to be creating a framework to evaluate the safety of bioactive proteins, especially recombinantly expressed bioactive proteins, so that industry folks like us at Helena and others, there's other great companies working on addressing this as well, can really consistently and reliably have a toolkit to understand, is this protein safe, and is this something that I can put into consumer products to advance these products and move this entire field forward. So there's really kind of 4 categories that we've thought about. The first one is reproducibility. So we need a framework that helps every single company and every single, not just company, scientist within the world of bioactive milk proteins to produce data that is consistent across several different types of proteins. So we want to be able to create a framework that doesn't just address the concern of one protein, but can be really something that's higher level that can be used for several different types of proteins that have bioactive properties. And again, this is like best case scenario. We would love this. Whether that's achievable or not is something that we all have to work together to get to. Something that is affordable. So we need to be able to empower researchers and businesses from, you know, the Helenas of the world, people like us that are still fairly small but growing, to, you know, large industry players, to be able to evaluate safety in a way that is still affordable and allows us to move the science forward without such huge cost hurdles. Timely, so, you know, studies should be achievable in a reasonable amount of time and empower industry to continue innovating. If we add years and years and years and years to the timeline of getting a new bioactive protein to market due to over testing, then that's going to really stifle the industry from moving forward. And there's so much opportunity here to move forward. So we have to be thinking about how long things take. And then we need general recognition. So everybody here today, and, you know, everybody within the scientific community who's really focused on bringing more bioactive proteins to our food supply, we all have to agree or kind of align on what this framework looks like and how we meet the standards of the scientific community at large. So these are all the things that, I mean, this is the stuff that keeps us up at night, that gets us into work every day, that we're excited to be addressing here at Helena. And I look forward to hopefully during the Q&A and afterwards speaking and connecting with many of you to figure out how we build a framework to evaluate the safety as we are able to scale this type of technology and bring bioactive proteins to consumers. So that's it from me. Thank you guys so much for allowing me to come in and speak today. And I look forward to joining you for our Q&A later.
Thank you so much, Laura, both for giving an outlook onto the future of maybe new ways to produce milk proteins, but also the challenges that come with that from a regulatory point of view. Really exciting. So I already saw many questions coming into the Q&A, so please continue typing your questions there, and we'll try to organize them into a logical order for the Q&A session later. But before that, I would now like to introduce to you Roland van Eerten. He's working on early life nutrition at Friesland Campina Ingredients, and he will give a lecture entitled Lactoferrin Development, Potential Growing Pains Unraveled. Roland, please take it away.
Many thanks, Casper, for your introduction, and thank you for the invite for this workshop. My name is Roland van Eerten. I work as a technical sales specialist within for lactoferrin within Friesland Campina, and I would like to explain you on practical hurdles we have to overcome when you go from a lab scale and pilot plant scale, small scale developments, all the way up to a commercial scale of lactoferrin. So like I mentioned, my name is Roland van Eerten. I work at Friesland Campina. First of all, I would like to give you a kind of agenda where I want to talk about for this afternoon or morning. First, a small introduction. Who is Friesland Campina and what is actually lactoferrin? Already the previous speakers were talking a little bit about this. Next is how do we produce this? What are the product properties of this lactoferrin? And then going more into the details, if you go from a lab scale, pilot plant scale, all the way up to a commercial scale, and from a practical point of view, where do you need to focus on and think about already at lab and pilot plant scale to make your commercial launch successful? So first of all, we have our anniversary actually this year. Friesland Campina exists already for 150 years. We are a merger of Friesland Foods and Campina back in 2008. Yeah, and this year we are celebrating our 150th birthday. We are an international dairy company, consists of 4 different business groups, having fresh dairy in food and beverage group, trading, commodity products like butter, milk, and cheese. We have a specialized nutrition business group. There we are selling infant nutrition. And last but not least, the ingredients business group. This is a business-to-business organization where we are selling nice dairy ingredients towards not only infant nutrition, but as well towards medical nutrition. And now diving into the details on the lactoferrin. What is lactoferrin? Well, lactoferrin is actually a protein which is naturally occurring in human milk and cow's milk, like explained already earlier. But only in cow's milk is 10 times less than in human milk. And therefore here in Friesland Campina, we would like to build or make lactoferrin, which is possible then to add as a fortification into cow's infant milk products to come to the same levels as what you would expect as well as in human milk. Why have we chosen for lactoferrin? Well, it has a high affinity of binding iron in the product or in your gut, so that creates as well as a support for the baby to their building up of their immune system and to reduce the risks of infections. We have produced this product already 30 years ago. We have more than 25 years of experience, and we were the first ones having this product with a novel food approval. But yeah, like I mentioned before, this is already a known technology. We would like to have a new technology, upgraded technology in the near future. I will explain you later about that. How does this product actually look like? So we have a highly purified protein product actually enhanced with a high level of lactoferrin with minimum 95%. These parameters are the most important parameters actually to think about if you look into lactoferrin as well. To maintain the bioactivity in this product is important, and therefore we have selected a process described like this, where we have we are extracting lactoferrin from fresh milk via chromatography. Via filtration, we further are concentrating up the lactoferrin in the solution. And after drying and packaging, you get this lovely brownish pinkish color, which has still some iron in it, so therefore it has this color, and which is then was possible for dry blending applications at infant nutrition. Like I mentioned, we produce this already in Veghel, and as well we have a facility, a joint venture in America, the UDAD in Arizona. But we have decided to build additional capacity as well in Veghel, and to learn actually to get the best of both worlds from Veghel and UDAD, and to learn as well with the new knowledge out there to have a more up-to-date state-of-the-art factory in Veghel. So that's for the end of this year, we would like to have this this new factory completely validated, and we would like to supply the lactoferrin as well worldwide. So coming more then into the topic of today, what does it take actually then to bring an idea, different new technologies from lab scale, pilot plant scale, to implement it into a commercial scale? How can we optimize the yield, which we currently have in Veghel and UDAD, and to improve the yield into a new factory? And this takes a little bit of time. Well, you don't design a new factory in a day, like you don't have the city of Rome is not built in one day, so it takes time, and you need to design your new process in correspondence with the product quality you would like to achieve. So there are many different choices you have, and it's like a Rubik's Cube. You need to twist and turn to get the most optimal process design and the most optimal product quality. They go hand in hand. Now, what I would like to explain more into coming few slides is then what is exactly your product quality, how to define this, and next to that, how to define then the process design next to it. So here I explain more about the product quality, and actually the definition of your quality is set by your user requirements specification. So this is a table showing, okay, what are the most important parameters you need to have in mind to make sure your product is successfully launched, that the customers are interested and are happy with your new product, that's according to legislation, that you can analyze it in the correct way so that your analytical methods is conforming the compliance, that you have set the right recipe design, using the right process additives, that your supplier management, quality management is set up right, that you have the right raw materials, you have the right quality of processing aids, the right packaging, because packaging is important as well for having the product safe during whole shelf life. If you have guaranteed your incoming raw materials and you have a stable process, your product quality has a higher success of stability throughout processing. So what I would like to explain is more into regulatory requirements, like already mentioned as well by the first speaker, which is becoming more and more important. What did we came across? And as well a little bit more on the product qualification and the validation. Where do you need to think of from a practical point of view to make your process, new process a success? So first of all, you need to think of what type of regulatory requirements are there. So if you want to produce worldwide, you need to take a look into EU legislations, in FDA, draft dossier, you need to have a GB legislations. You need to think about, okay, which parameters are needed to be able to be active on that market, and not only looking into your raw material, but as well looking into the requirements for the final goods. You need to take both into consideration. Then what we had, for instance, when we were running pilot plant trials, we were using certain processing aids and additives which were actually not allowed to be used in the lactoferrin when we are going to produce it for commercial in different regions of the world. So already in lab and pilot plant scale, you need to think ahead. What type of processing aids are you able to use for your product if you are going live for in China or in the US? You need to think those requirements already ahead. Next to that as well, how to analyze your product. As well, different regulatory requirements can be out there for your for how to analyze your parameters, and you need to understand already in advance what type of methods you need to change or take to make sure that you are compliant to the region you want to sell your product to. Next to that, if you are looking into a product qualification and the validation step, important already at the pilot plant lab scale is that you know which parameters do you need to focus on if you want to start up your new commercial process. So which ones are the most important to test and to steer already your commercial process so it will become quicker a success in commercial allowance. Next to that, you are working quite heavily in your own project. Eventually, there's an analytical lab who needs to analyze all those different parameters. So you need to involve the analytical lab as well as soon as possible. If you have set up the whole URS specification, take your analytical lab as well into the discussion so they understand their urgency and as well meet the time requirements for the project to make it success. What we have learned in the past is that if you don't involve an analytical lab early in the process, then it can take some more time than you wanted to get the results back, and that in order to steer the processes in a better way. And last but not least, if you have as well new customers coming in, you want to make your product in such a way that the customers accept your product is according to their expectations. So it's not then looking only on nutrition level, micro level, or contaminants level, but as well, for instance, on powder properties. Does your product flow correctly? Can you dry blend it correctly? And do you need to tweak and turn there something to make it worthwhile for your customer? This is more on product quality. If you now look into processing design, we get a lot of information from an R&D. So at the lab and pilot plant scale, there's a design book written. And this is actually the start of setting up your engineering work to scale up your product. When you have, you start to build your factory, and then afterwards you start to validate your process via an IQ or an OQ or a CQ, the cleaning qualification, to make sure your process is designed in such a way that it can produce your product correctly within specifications. Next to that, of course, your factory, in our case, is part of the Veghel organization. So there needs to be a good interface between the project team and as well the project location, because eventually your production location needs to hand over, needs to take over your processing line. So they need to be early in the project to understand how to steer your process in such a way to make it stable. And last but not least, ensure a stable process is needed. It's not something you think about perhaps already in lab and pilot plant scale, but it's something you need to think of in commercial scale, that you are able to produce not only a product once in a good manner, but it works 24 hours, 7 days a week. So I will explain a little bit more about the scalability and the stable process. What is most important in scalability, what we have learned is that you need to translate all the knowledge you have obtained from pilot plant process towards a commercial scale. For instance, what we went into is that when we were doing the lab scale analysis and the pilot plant scale analysis, this was done for instance, with a pasteurizer, with a laminar flow instead of a turbulent flow. So if you are building a commercial scale factory, you need to think as well on those type of small details to understand that your product can act different in a laminar flow or a turbulent flow. Your effect on time temperature combination in a pasteurizer will be different. Furthermore, something to be taken into account as well into a commercial scale is that you avoid your post-contamination. So how design your factory in such a way via sampling and CCP points that you make sure your factory is designed in such a way, including the right cleaning conditions, that you don't have any post-contamination of your product. Looking into stable design, what we have learned from our lab and pilot plant work, it's already important to pick out the right parameters which are most important for your process to steer, especially in the commercial ramp up. When you are focusing on all the different parameters, it will be very difficult to ensure a stable process. So you need to focus on the process parameters which are most important for you to steer the right quality parameters. Coming to a closure, so what are my take-home messages for you as well when you are developing new processes going into commercial factories? So take a regulatory landscape already earlier into account, already in a lab and pilot plant work. Take into consideration what is not only giving you a stable product, but as well what is from a regulatory point of view, how to make your product from a regulatory point of view in the right way. And be flexible as well from process and product design, because there will be changes during your whole project from regulatory, from analytics, from customers who are interested but have different requirements to your product. You need to be able to take this up into your process design. Eventually be as well pragmatic and think about your customer expectations, not only on the ingredient itself or the product itself, but as well the impact on your applications. So how does it eventually dissolve, or how does it eventually dry blend in a powder product? And that was my presentation.
Thank you so much, Roland, for this very interesting presentation. After some talks mainly looking towards the future, it's also nice to see how things happen in practice with a more retrospective point of view. So thanks a lot for that. Next up is the final and a dual presentation by Anna Stout-Quistgaard and Peter Weesche, both from Arla Food Ingredients. They will jointly give a lecture entitled Building a Bridge from Past into the Future for Food Ingredient Commercialization, the Story of Osteopontin. Please, Anna, take it away.
And I was hoping that my co-host, Peter Weesche, would be joining in here.
Do you hear me?
I can hear you, yes, Peter.
You hear me? Okay. I don't know why the video is on, but seems to not work.
Can you see the presentation, Peter?
I can.
Okay, I will do the slides.
But can the meeting, do you hear me?
Yes, Peter, we can hear you.
Yes, they hear me. They hear me. Okay, so I can voice over. although maybe you don't see me.
We can see you,
Sorry
Peter.
about that. Great. Yes. So we're going to give you a talk here about some reflections over the development of osteopontin by Arla Foods Ingredients. And I can just have the next slide, please. And
Yeah, it should be coming.
It should.
Here we go. Sorry.
Yes, first the disclosures. So, well, and maybe you are from Arla Foods Ingredients, whereas I used to work with Arla Foods Ingredients and were part of the development, but now I'm currently employed by Arla Foods Amba, and I have no longer anything to do with osteopontin. But so I'm just describing something that happened historically, and then I'm part of the steering committee of IMGC. Yes, and osteopontin, what is that? It's a small protein, around 300 amino acids, that's found in bovine as well as human milk. It's a multifunctional and highly phosphorylated protein. It's implicated in a lot of different biological processes, and it's found in a lot of tissues and bodily fluids, with the highest concentration found in milk. It doesn't have a tertiary structure really. It's an intrinsically disordered protein. So that gives some advantages that I'll return to, but there are many proteins that are active due to their tertiary structure. But osteopontin's activity relies on its flexibility rather, and then due to the flexibility, it can interact with integrins. It has integrin binding sites and also calcium binding sites. And also by being that flexible, it's prone to protein, you can say degradation or modification. So that is probably part of the regulation and the reuse of the osteopontin protein as a motive to signal via the integrins. Yeah, but it all started a long time ago by the researcher Esben Skipper Sørensen at Aarhus University, who was looking for a topic for his master thesis, and he had that reflection that it should be something that's actually could be made available from milk. So what did he do? Well, he skimmed the milk, he boiled it, he added acid to it, and what stayed in solution would be somehow, you could say, both maybe easy to work with, but also something that could be easily applied. Yeah, what stays in solution when you do this to it? That's what's called the proteose peptone, and that was the topic he worked his way into. Now, next slide, please. What did he find? He found osteopontin, and that was only the starting point. Firstly, purifying and characterizing osteopontin as well as other proteins from this fraction of milk, and then looking into all the post-translational modifications, looking into what kind of function would it have, the biological meaning of this protein. And that's been a long journey and a long and fruitful career built very much around osteopontin. Next slide, please. Then at some point, his work and the likely possibilities in this protein to Arla Foods Ingredients. And so you can say what has then been worked upon in the middle, we have the purification and production of this protein. And then around, there are all the different potential uses of it for infant nutrition, for wound healing, for dental health, for immune health, even cattle fertility and bone health. So because it's a multifunctional protein, there were so many potential opportunities to apply this protein. And next slide, please. So Arla was inspired by Espen's presentation and started working with this protein. And the first thing was to scale up the production to be able to really, well, both see if a stable production would be possible, but also to have more material to work with. And that was, of course, quite a leap to go from milligrams and into kilograms of pure protein. But that happened around 10 years after Espen actually identified the protein in milk. Next slide, please. With the work happening both on the production process, but also on all the applications, of course, a lot was invested, both of human resources as well as money to support all the research. So to protect all that investment, patents were made by Arla Foods Ingredients. Firstly, on the production process and several variations over that, and then also for some of the uses, the potential uses of this protein. Next slide. So, of course, patents make sense to fend off competition and to protect the investment. But there are also some other consequences of patenting. When it's your own, it's also for yourself to promote it. But we will return to that aspect later. Next slide. Yes, but just to kind of give you an impression of the complexity of all the work that was spun around this molecule, in the middle here we have the osteopontin and the patent on the production process, and then all the different potential application areas and research projects, PhD projects, customer discussions, and so on happening over time, building and shooting new branches. By the years, it just grew and grew. And it takes a long time to take a new molecule all the way into recognized application. So a lot was really happening here over the years. Next slide, please. And on some point in time, this had to be tightened a bit. So there was a strategy formed around all these potential uses and also around the raw material. So on the right-hand side, you have the bovine milk-derived osteopontin. And it was concluded that infant formula as well as dental care were the most likely application areas. And then there was also a lot of speculation around other osteopontin types and what they could be applied for. But then from this time in history, the focus were on infant formula and dental care. Next slide, please.
And then I think I will be taking over, Peter.
Yes, you will.
I'm glad that you had the ability to join in, even though there were some technical challenges. Now, I will be discussing a little bit further about bringing osteopontin to the market. And as Peter just described, we went in many directions because we were dealing with multifunctional proteins, a multifunctional protein. So we really had the opportunity to go in many different directions with that protein. But at some point, we also needed to narrow the scope and focus our resources within the organization towards some sort of positioning and commercialization of the ingredient. Now, the aligned research strategy was for us exactly that. And we had defined to position osteopontin, or here you can see the commercial name, Lacodan OPN10, into the infant formula category. And the reason that that makes sense for us at that time was that, as Peter mentioned, there is a large content or high content of osteopontin in milk. It's larger in human milk compared to bovine milk, and therefore there's also a gap between the osteopontin level you find in human milk compared what you find in infant formula based on bovine milk. So you can say this represents an opportunity to close that gap, compositional gap between human milk and infant formula. Now, I wanted to just pause a little bit on the osteopontin story and talk a little bit about the infant formula, the early life nutrition segment in a more helicopter frame, so to speak. Now, infant formula category has developed significantly the last 50 years, really moving from a very basic composition consisting of whole milk powder, sugar, some added vitamins and minerals into the infant formulas you find on today's shelf, which contains highly sophisticated ingredients. Examples of that could be the long-chain polyunsaturated fatty acids like DHA and RNA, nucleotides, pre and probiotics, and so forth. So really a journey from something fairly simple to something what I would consider highly sophisticated nutrition. The science-based innovation within the infant formula industry are often driven by two forces. There is a compositional differences between human and bovine milk, and therefore also infant formula based on bovine milk. And then there are development differences between the breastfed babies and the formula-fed babies. Now, despite the high level of product development, I would say particularly the last 2 decades, we have really seen a lot of significant development within the infant formula ingredients made available to put into an infant formula. Despite this, when you observe on a cohort level, babies being breastfed versus baby being formula fed, we still see differences between these two populations, so to speak. And some of the elements have just been listed at the illustration down below on the slide. So we see differences in terms of growth, weight, immune system, infections, etc. So even though there has been this development, you can say these differences that you can keep observing between those two cohorts of feeding regime to at least to a certain extent represent a driver for innovation, and therefore also osteopontin put into that perspective. The regulatory part has already been mentioned throughout the entire session, which I think is really neat, because there's definitely a common thread throughout this conference that is also evolving around regulatory. I will, however, just talk a little bit about it because it's such an important area for getting innovations into the industry. Now, obviously, infants are the most vulnerable consumer group, and therefore is and should be highly protected by the regulation. At the same time, we're also looking into a fast-moving, you can say, technological capabilities that provides the opportunity to put these highly sophisticated ingredients into the infant formula category. Right now, we are looking into HMOs, so human milk oligosaccharides, being launched in infant formulas in several regions of the world, and also with the required regulatory approvals around that. So that's really, really interesting to monitor that road from innovation to a commercial ingredient and being placed on the market. So this means that, so if you compare the the technological advances, the possibilities, but also an increased regulatory barriers or requirements. We're definitely at a place now where obviously authorities need to identify the proper route for approvals, and obviously companies need to identify how to work with the authorities and deliver the data that are needed, and having those discussions with authorities about what is the right safety assessment. I think it was Dr. Tomei who opened up about this is really happening on a case-by-case evaluation and assessment by the authorities. And I think it's so critical for the industry to be in close dialogue with the authorities and really navigate on a case-by-case level of the different potential innovation and how to get the proper safety assessment of these. Now, moving back to talk a little bit about the commercial ingredient, Lapodain OPN10, and bringing that to market, that obviously required significant resources going into multiple directions. Even though we sort of narrowed the focus on infant formula category, there were a lot of elements that needed to fall into place. There were research done into the osteopontin production. Peter showed that picture of going from milligrams to kilo, and obviously a lot of research was behind those advantages. We also needed to look further into how do you analyze it, and I think that somewhat reflects also what was being discussed on the previous talk on lactoferrin. So getting those analytical methodologies in place, being able to analyze both in human milk, bovine milk, and infant formula. There is a lot of preclinical evidence and obviously also clinical safety and efficacy evidence that will be a part of bringing such an innovation forward. And then, of course, the regulatory approval, which is a significant and impactful category that you need to strategize around. And the fact that Arla Foods Ingredients has protected both the production process and an application process of osteopontin, that definitely puts us in a position with a lot of opportunities. But you can also say it also has some consequences in terms of us driving all of the above, where if you look into the lactoferrin segment, there are multiple producers of that ingredient that could represent opportunities to do some collaborative work. One area where we needed to do more work once we sort of had focused down that it's the infant formula category we would like to position osteopontin, or at least investigate positioning of osteopontin into, was understanding the level of OPN in human milk. When we first started out on this journey, and back in the early 2000s something, we had the initial data of 18 women, so osteopontin level of 18 women, and that was sort of our initial platform in terms of understanding what is the gap between human milk and bovine milk and infant formula. So we definitely needed to get a better grasp of what is the OPN content in human milk. And in order to do so, we joined forces with 3 other partners and analyzed human milk samples in different geographies or different regions of the world, and also at different lactation stages. And you can see here that we had a total of plus 800 human milk samples from plus 600 mothers collected in this survey. The results of those analyses you can see here. We did see some differences when we were looking throughout regions. So definitely a higher OPN level in the samples coming from Asia compared to what we found in Denmark or Europe, if you will. And we also identified a decline of OPN throughout the lactation stage, which you see with many other components in human milk, where certain levels of proteins, bioactive components will be fairly high in the first week after giving birth and in the early stages of lactation. You'll see this, for many components, you'll see this slope falling down throughout lactation until you reach a level of what you can call mature milk. In addition to the work on analyzing human milk, we had an extensive work laid out in front of us. And here you can see the main part of our study portfolio. The portfolio included toxicology studies and various animal studies. Here we were looking into vaccination responses. We were looking into intestinal development, intestinal scriptomics, all to investigate how does OPN impact the intestinal tract. Here's just a very short overview of some of the toxicology studies. And what is good about that part is that it's fairly stable elements that needs to be done for regulatory approval globally. Finally, we engaged into an infant clinical studies with a partner. That study took place in China and consisted of OPN-enriched infant formula compared to a standard control group and a breastfed group as a reference. Obviously, the breastfed babies are the golden standard, which we're always reflecting any nutritional and intervention towards. The study as such concluded that the OPN-enriched formula was well tolerated and supported age-appropriate growth. We're coming back to the regulatory landscape. All of these efforts were just awarded as we received a positive opinion from EFSA or the European Food Safety Authorities for the infant application of Lacoda OPN10 just about a month ago. So we were very excited about that. Now moving towards learnings, and you can see this dot saying 93. And that's many years ago, but that goes all the way back to Espen and his initial work at Aarhus University. But it's definitely some of the key learnings is we would like to highlight here is about a more targeted approach in our research strategy, defining the core and invest into that, and maybe only invest into that. Get your analyticals in place. Those are key. I think that was mentioned for the lactoferrin as well. Get the human data in place, doing in a highly quality manner. The toxicology package is just a must. It's just a must for whatever you position your ingredient forward. And then, of course, securing infant clinical safety study or data in a high quality safety study, engage with authorities, and also secure the regulatory approval. And for the US, those data needs to be, or US approval, those data needs to be made publicly available. So that's also part of the timing, but also your potential IP consideration. So this is my last slide. Something about BECBID. Do they invest ambitionally into the strategic area and be aware of the changing conditions over time? At least the regulatory landscape has changed significantly the last 10 years, and barriers are increasing with further levels of evidence being considered and requested by the authorities. And when you sort of compare the academia setting versus the industry setting, then we're definitely operating from a different time scale and goals and measurements. So that's also something to consider and reflecting the points above. And I think I'm being out of time now.
Later for this wonderful presentation. So after we have all the presentations, we will take a 5-minute break, after which we'll come back with the panelists for the discussion based on the Q&A. I already saw many questions coming into the Q&A, but if there are any things that are popping up in the next few minutes, please write them there and we will deal with them in 5 minutes from now. So at 1 minute past whatever hour you have close by, we will reconvene and start again for the panel discussion. So hope to see you all in 5 minutes. Okay, everyone, welcome back. I hope you had a nice short break so that you are nicely refreshed for this final hour of today's workshop, in which we'll have a panel discussion. And as already mentioned, questions in the Q&A are used for that. So if you have any more questions you would like to discuss, please write them in the Q&A and we will try to incorporate them. But to get started, I thought I heard or I read something already quite early on in the Q&A, which is about what is your comparator? Because if you look at human milk, there's a lot of variability. And that was actually also already picked up in the last presentation on osteopontin, where some data was shown on how much variability there is between individual months and over lactation in the level of osteopontin. So I was wondering whether someone would like to comment on that. So how much of a component should be added if you want to mimic breast milk? Should you take an average value, a minimum value? What's safe and how to get approval for that from a regulatory point of view? So who would like to start?
I can definitely start if you allow, but I'm sure the other panels has some super good insights as well. But it's definitely touching upon a big challenge for everybody, both from the baby's perspective, you can say, all the way for regulations and companies producing an infant formula, because what is that right level? And you can say one of the challenges is that we have this division into stages. So there's a stage 1 and a stage 2 infant formula. At least now I'm talking from European perspective. I'm not sure how it is in the US. Anyway, but where the infant formula is from 0 to 6 months, that's the stage 8, and that's obviously targeting right into the lactation stage where human milk is evolving significantly and changing significantly. And one perspective, and I know there's been done or has been considerations about this, is that staging, that further staging of infant formula, where it could be an opportunity to look further into maybe narrowing the brackets of when an infant formula, at what stage or age point that is applied. That could definitely be an opportunity, because then you'll be able to target even further this level of concentration for that stage. It does represent a lot of other challenges, and I understand that those challenges both from a regulatory point of view and a consumer level, but just part of pointing into that direction, that will definitely make it easier to have more targeted or narrow stages around the infant formula. That was my 2 cents.
Thanks, Anna. Roland, maybe you can also say something about how this was dealt with, and then I will go to Daniel for the...
Okay, I can. Okay, I just want to comment. It is really an important concern, this problem of which quantity can be used. We should not forget that we are in the food area, not in drugs. And so it should be what we could consider as a dietary quantity. That is the first problem and the first question that is addressed by a regulatory organization. And so an important information very often is to start by the quantity of the component that is traditionally consumed from traditional food. In some cases, it is known. From experience, in other cases, we really don't know. For instance, I remember in Europe, we had a lot of discussion about lactoferrin. And to know which quantity of lactoferrin is consumed by traditional food, dairy food, and we just realized what very often we don't know what is the quantity of lactoferrin, for instance, in cheese. Is there any lactoferrin that remain in cheese? There are some results that show that in some cheese it could be a lot of lactoferrin, but in fact, it's not very precise and we are not really sure. So I think probably the first step would be for an applicant, for instance, to try to know what is the quantity traditionally consumed. After we can discuss how many times we can increase this quantity, 2 times, 3 times, 10 times, I don't know, it depends on the component.
Thanks, Daniel.
To add on that, yeah, I think your golden standard is the mother's milk, and it varies a lot. At colostrum, you have much higher levels of bioactive components than when a baby is already 6 or a year old, 6 months or a year old. So it's difficult to justify what would be the levels you need to test. I would expect there's some evaluation you do already at clinicals or at in vivo testing to see, okay, to which levels do you already have a positive effect for babies, and how much do you need to add then into your infant formulas to make sure your babies do have this positive effect eventually when they consume it.
Thanks for the addition, Roland. Then there was also a question that's somewhat related to this, and that's not only there is variability in the human milk as such, but there will probably also be variability in digestion. So this was a question which was asked during David's presentation. So actually what's finally ending up is not only a matter of what's consumed, but also what's happening in the digestive tract. David, how do you look at that from what you have heard up till now?
Yeah, thanks, Casper. So yeah, and I think so it means the person is right. I mean, there is variability across digestion in terms of who the consumer is. You know, we see digestion differences between preterm infants versus term infants, and that will change what milk is delivered, like what form milk is delivered to those babies. And then we'll probably see changes across time of day of life as well. And then, of course, there are variations in human milk itself between mothers. So all of those things have to be taken into account and studied. And then sort of to the question, I think in terms of like what the gold standard should be, someone mentioned in the chat about mother's milk versus donor milk. And I think in this case, we need to use human milk, mother's milk that's not processed, that's raw, because donor milk, the heat processing that's used really does change a lot of the bioactive proteins that are present. So it may not be the optimal comparator for trying to determine what's the best levels of certain bioactive proteins. Yeah.
Thanks. And another type of variability, to stay within that theme of variability, is also, I think, therefore, recombinant proteins. Because if you look at recombinant proteins, you try to mimic as much as possible what's present in human milk. But with precision fermentation, you may not have 100% identical post-translational modifications. So, Laura, how do you look at that? Is your target really 100% identical, folding identical modifications, or what's the target in the end?
I think it's possible. It just comes with a lot of like engineering. I mean, you know, what we do at Helena is glycoengineering. That's really the core of our focus, because we know that the glycan profile of specific glycoproteins in human milk, it's important, and we don't want to be producing any glycans that are not safe, which, I mean, there's plenty of data out there that, and, you know, partners that we work with to figure out, you know, the glycans, but we can be quite targeted. I think, you know, something that we have to come to terms with, I mean, we have, and I think the industry does as well, is when, you know, certain proteins are secreted into breast milk, there's so many different glycans that we can see on a protein. And so we're not going to be able to mimic all of them, but we can at least capture one glycan profile. And we know that like babies have been exposed to that. So that's really the strategy. And I think that trying to achieve something as close as possible through microbial expression is something that we can work towards as an industry. I mean, tools like CRISPR, which is something that we use, make it much more viable than even 5-10 years ago using different techniques. So I think it's a challenging goal, absolutely, but it's possible, and that's what we're working towards, is 100% identical to the native.
Yes, thanks a lot for that. Then besides variability, there were also several questions that all relate, I think, to the concept of synergy between proteins, because some people ask about proteins may work together, or there may be matrix effects which actually affect the functionality. And if you are testing outside the milk matrix, do we still see the same thing as when you would just consume milk? Who would like to comment on how to deal with that? So how to deal with synergy?
I mean, I can speak on the matrix effect issue. One thing that pops in my mind is that human milk also has a lot of different enzymes and proteases that could affect the survival of a protein. So that could be quite different than a formula that may not have the levels of those enzymes present. So that's one thing.
But I was also, I actually wrote down this question myself already when I was listening to you, Daniel, because how do you deal with this from a regulatory point of view? Because if you know a specific component, you can measure it, you have a benefit, you measure the benefit. But what if the component is actually a complex of components and there is not one very specific benefit in the end, or there may be multiple ones? How should you deal with that from a regulatory point of view?
Yes, this is a very complex question, and very often for the regulatory aspect, one of the main difficulty to provide clear answer. For instance, it's like drug. When there is a very specific component, an isolated component, well characterized, and so on, it's easier to evaluate only the effect. We know that the component is clear. When it is a complex mixture, we always know that it cannot be completely described. We had very often this problem with dairy product, but also a very good example is also many plant-derived products. There are many molecules. Even, for instance, if you look at the composition of the soy protein isolate, it's always about 90% protein. What are these 10% which are not in the protein fraction? And as there is many claims which are asked now related to soy protein products, it's a big problem to know whether the activity that is shown, in some case very relatively interesting activity, is it due to the protein fraction, or is it due to the non-protein fraction that remain unknown? So I agree, it's a very, so in some case after I say it is a kind of politic decision. If there is absolutely no deleterious effect, that the positive effects are well demonstrated, the technology is relatively well described, the source of the product is well described, in some cases it is accepted. But I agree, it's a big difficulty.
Could I jump in quickly?
Yes, please.
I'm totally blanking on which company proposed this to the FDA, but there was recently a proposal for the use of, I think it was something between 3 to 5 different HMOs in a specific combination to be used in infant formula. And so I think that's a really interesting example of components or, food ingredients, HMOs that all have varying different bioactive properties, as HMOs do, all used in combination in an infant formula. I don't know if anybody here, any of the attendees have seen this, but I was recently reading about that, and I think that's an interesting example touching on this theme of like synergies of different things. And I think that there is progress being made, at least from the industry perspective, of combining different ingredients that have different properties.
Thanks. Anyone from the Friesland Campina Arla point would like to comment on how they view this synergy and how they deal with it from their industrial perspective?
It is definitely a complex issue, and I don't think anybody have the straight answer or clear answer on how to navigate within that. You can say that the final testing in terms of safety would be that infant clinical safety trial, which will reflect, at least from a safety perspective, synergies or its safety together with other components. But once you go into the toxicology package and maybe some of the animal studies or other preclinical studies, then that synergistic effect can be very difficult to analyze and fully get a grasp of, because there are some limitations both within the animal setting and in vitro settings where you're not capable to get that full perspective. So unfortunately, I think the infant clinical safety trial will be that one that provides the insights into that final matrix. Yeah.
Yeah, if I can add on that, it's already difficult enough to quantify the bioactivity of a single ingredient in a dairy matrix. So what we learned already is that there's development ongoing on bioactivity for lactoferrin, for instance, in the infant formula, and not only then looking on the quantity of lactoferrin being analyzed, but as well what is the effective lactoferrin still in this product. So you see there's movement, but at this moment it's really in child shoes, and start in small steps. We are developing a more understanding from a global point of view how to analyze then the bioactivity then in a final good. You see that from a regulatory point of view, but as well how to analyze them eventually, that everybody within the whole world is able to analyze it in the same way, and that you get the same result. So I think it's important to justify addition of bioactive ingredients in your product, that you can test it. But yeah, we are not there yet that there's an AOAC method who can really give you the answer on bioactivity already for only one active ingredient in a recipe.
Yeah, and you can also say that what synergies, and should that be maybe formalized into at a different level, because Laura, you pointed out the multiple HMOs being tested, and that's super relevant. But what about then the other components, the nucleotides, the LC-PUFAs, the lactoferrin and the OPN together? What does that, and that's sort of where you're sort of left up your own devices on how you're going to benchmark towards what, and what synergies are you going to be investigating. And from a regulatory perspective, I could imagine that's hugely challenging to be navigating within that field and trying to set directions. So for this ingredients, we need to know A, B, and C, and for that ingredient, it's a different direction. Yeah, so very challenging.
we've been talking a lot about infant formula, but what about the applications in other populations like elderly, medical foods, athletes, toddlers? Like there is a whole like lifespan, right, that we can address. And how does the regulatory, how does regulatory treat all of these different bioactives and their functionalities?
I can tell this for the different population, the rule is to provide results on the target population. So if you want to develop complementary food, for instance, for older people, you have to experiment in a group that is representative of the older people population that you want to target. I agree, it's a difficulty very often because according to the population that is targeted, it's not so easy to experiment. Or you can demonstrate that let's say the mechanisms are the same, for instance, in younger and older subjects for one specific component. So if you can show this, I think you can provide result on younger subject that is always easier than, for instance, older subject. We know it's a very difficult population to experiment. But anyway, the rule is to show that your experiment can be extrapolated to your target population. After, it's not a problem to use a pure ingredient or not. After, it's a safety question. To have an evaluation, as I say, of the usual quantity that is consumed traditionally, and probably if you propose to introduce, I don't know, more than 100 times the usual consumption, it will be probably a problem, because we'll say we are not in the diet, in the food area, then you are going to the pharmacology. But always the balance between two.
As a follow-up, Daniel, how do regulatory agencies decide on the outcomes that they're going to accept as important? For example, say a bioactive increases weight gain in infants, but it increases gut inflammation. But gut inflammation isn't a standard outcome measured, let's say, right? So where does the FDA and EFSA, how do they decide on the outcomes that are important and what's acceptable? Because we want to make sure we're not introducing a problem in one direction and improving a different one, right? At the same time, we don't want to create problems.
Yeah, it's always the question. That's what I wanted to show. For instance, for the claims targeting the gut physiology. For instance, we know that the first thing that is evaluated is whether there is an effect of traditional gut pathology like the diarrhea. And in some cases, we know that some components after a certain level of intake are going to induce diarrhea. And so this is the first question. So I think diarrhea, after you have the problem of allergy, for instance, it's very often some components ingested into high level can induce allergy, particularly when it is a protein. For instance, we know that so it's not allergy, but it was shown not for lactoferrin. It was shown that people ingesting for a quite long time a high quantity of lactoferrin of bovine lactoferrin produce antibody for bovine lactoferrin. So we could have some concern about, in some cases, can it induce allergenic antibodies. And it's not the case with human lactoferrin, because
something, sorry, Jennifer, just to jump in, if you don't mind. I think something that's important to consider when you talk about the other applications outside of infant formula compared to the infant formula application is just how much of these proteins are people actually being exposed to? With infants, I mean, formula is the sole source of nutrition, right? And the regulatory concern for establishing safety and understanding efficacy is going to be completely different than if you are exposed to small amounts of this protein and you eat them, you know, a variety of so many other foods. Like, how much of an impact is this going to have? Or I guess more of the question is, how much of a concern do we need to have when this isn't part of the sole source of your nutrition? And so I think that, you know, as we see it, there's a world in which, you know, likely you put these types of proteins into non-infant applications first, because there are, you know, there's just a completely different risk profile in proceeding in that way before putting it into infant formula. I would say particularly recombinantly expressed proteins and particularly human milk proteins as opposed to things derived from bovine. But I do know that that's a really important consideration. And I would anticipate for regulators is also, at least in the US, is what I'm more familiar with, is also something that will be considered when these things are evaluated to include in foods.
Thanks for the addition. I have another question to you, Laura, about also a bit this point of synergy, but then if you apply precision fermentation, you have to select a specific target protein. So are you also taking into account there this possible synergistic effect of human milk components? And if you look at the whole range of proteins you can find, you are talking about hundreds to thousands of proteins. So how are you taking that into account in deciding which proteins to focus on?
That's a great question. I mean, to build a viable business, we had to like start and say, okay, here's what we want to build. So we have looked at the way that we came to at least the first protein that we're working on is really what's publicly available and understood. As we've learned, I think through today, is that, you know, for a protein like osteopontin, we need to really understand how it is the variability in concentration look between parent to parent, between different stages of milk being produced. And that is, you know, always going, or will continue to be, the question for so many proteins, but we've really focused on proteins where we believe there is a good amount of available data on the composition, the quantity in human milk, the consistency from parent to parent. That's how we've narrowed down, because at the end of the day, for us, as we think about commercializing a food ingredient that is a bioactive human milk protein, and that will be something that we want to use in infant formula and other applications, we need there to be some kind of general recognition that this in and of itself is safe, and that there's general recognition from the scientific community about like what it means to create this protein and what the profile should look like, and what, you know, from there, what kind of safety aspects to look at. So that's been our focus, at least to start. I think that there's a lot of really fascinating proteins in human milk that are maybe there in smaller quantities, or things that we would like to get to eventually, but requires probably more studies on, like, just compositional studies on human milk to understand better. So that's been our approach. And, you know, at the end of the, we're creating a consumer product. So we want to be able to make something that will be suitable for as many infants as possible. So it's just a little bit of a different lens that we look through when we make decisions on like what to work on and what to develop.
Thanks. Very useful to get those insights as well. Then a question that was asked about lactoferrin, but I think it's equally valid for the osteopontin case, is if you are selling a certain functional protein, are you, or maybe should you also test functionality of every batch that you are producing? So how do you perform quality control on the product that you are delivering?
Well, at this stage, it's not requested by regulatory to have as well the bioavailability tested for every batch you are producing, but we do see a movement that active lactoferrin is important to analyze as well on final goods. So we see now customers asking certain more requests on different analyses than what we currently are doing as well to test the functionality of our product. And yeah, this field is rarely new, like I mentioned before. So we are discussing as well. So how does this regulatory landscape will change, and as well how do we need to update then our methods, or maybe create new type of methods which are as well worldwide and validated in order to analyze then the bioactivity. You can analyze bioactivity in many different ways. So which type of method do you want to use to quantify then your bioactivity? And I think that will certainly come, because it's important. If you add something in your final good, you need to make sure that it really works and it has an added benefit for babies. So I do believe this is new ground, new territory, which will be further developed. And how it will be developed, that depends a little bit as well on how authorities are developing their regulation, and how as well our customers are implementing these new legislations, and how we as an organization of product developers, of dairy manufacturers are able to test those type of products on bioactivity. So it's something ongoing, most probably.
But do you think that it will be driven more by legislation or more by the customers buying your ingredients?
That's a good question. But what we notice is, first there's a drive from regulations. As far as I can see, it's not really requested by the mothers or the fathers who are giving the infant nutrition and to their babies.
And I can add, you can say there's of course multiple layers of parameters to evaluate. One thing is when it leaves an ingredient supplier like Arla Foods Ingredients or Friesland. The other level is what happens when you apply it into an infant formula and processes it further and then out to the final consumer. But definitely super interesting and relevant aspects, and something that I think we definitely need to investigate further into and understand that how does processing, whether it's from an ingredient supplier perspective or final infant formula, how does that impact what's out on the shelf in the supermarket or at the mothers and parents or the parents' kitchen and feeding their baby with. You can say from an ingredient supplier perspective, it's definitely surrounded by extensive characterization of the component and quality analysis being applied also for the commercial products. So obviously both in the research phase, developing phase, and then into the commercial production phase. So extensive characterization both on chemistry and quality parameters. And then you can also add that that the trials that you have done will be done on commercial representative samples. So somehow there should be a link on what you have seen in your various trial. If those are done on the commercial, either commercial ingredients or commercial representative production method, that should be reflecting what it is that you are selling.
But as I said before, you cannot communicate anyway if you don't demonstrate on the human target population the reality of the effect that you want to show. So and so it's probably now more and more a very big concern for all the food company because it's more and more for ethical reason. It's more and more difficult to experiment first in animals, second in humans. And so probably there is a need to work probably very rapidly to develop non-invasive method in humans. What can we measure, for instance, in infant that is non-invasive? We can collect saliva, blood, something is very difficult, air, I don't know. So you really have to develop non-invasive method when you want to show an effect in infant, also in adult. It is probably one of the priority now for the more to have claims and to demonstrate specific effect of foods. Because we know that in the future, it will be very difficult to experiment. Particularly for food, because we know that if we say to the consumer, we collect blood sample in, I don't know, 100 infant to show that this formula is better than the other one, forget it.
No, but regarding these claims, there was also a new question that was just asked that is about claiming products to be like breast milk. But if you look at the complexity of breast milk, how should we look at such claims? And can you ever claim a product to be really like breast milk if you don't have that full complexity of breast milk included?
Just a short comments. I think the gold standard is not breast milk. The gold standard is breastfeeding. Very different. It's all a relation, food, feeling between the mother and the baby. And I think it's unrealistic to want to mimic the breast milk. First, we are not sure that the breast milk is the gold standard for all the components. For instance, the breast milk is deficient in vitamin K, it is deficient in vitamin D. In some cases, there is too much vitamin A because the mother, for instance, consumes a lot of beta-carotene or retinol, and then it can be toxic for the baby and so on. So I think it's a very difficult question. I'm not convinced the good way is to try to mimic the breast milk. We can look what is in the breast milk. For some component, it's probably very interesting. Lactoferrin is a good example, but also for the protein content, it's sure that we have to adapt protein ingested by the baby to the protein content of human milk. It is very low and so on. But I'm not sure we have to try to mimic for all the components. For many components, the composition of the human milk is a picture of the composition of the blood of the mother.
I like your comment here, Daniel, and I agree completely. And that's also when if you're looking for a driver of innovation, composition is definitely one of them. But I agree that when you then look at the different development patterns that you can observe, then that part is only a piece of that puzzle. It's also the you can say the feeding of the baby, the interaction with the baby, all of that sort of comes with the breast milk that you can't control for in a setup. What does that contribute with versus what's in the bottle or in the breast that's actually feeding the child? I think there's multiple facets around that we still don't have a good grasp of, and there you can say one perspective is definitely to look into the human milk composition and trying to identify relevant gaps, not all gaps, as you say, and that will not be possible either, but relevant gaps. And then I think it's probably also a part of the legislation. Because one thing is to look at it from an innovation driver perspective. Another thing is that what is being communicated to the consumer. And I think we as an industry can be allowed to look at this from inspiration, innovation drivers, possibly areas where we can improve. But it's definitely a very different dimension if we're talking consumer communication, which luckily is in many parts of the world is very much protected and regulated on what you can actually say. But I think there are infant formula cans out there where it says something about mimicking human milk. So yeah, probably we'll see a tightening up of that aspect moving forward. Yeah.
Then I also have a question going a bit back to science, and that's related to your presentation, David, where you mentioned that there are actually also a lot of peptides present in your samples, either initially or after digestion, where we have no idea yet about their functionality. But if you have so many of these peptides, how would you ever go about screening them for functionality? I don't guess you want to do that one by one for all the peptides that are there. So what's your view on that?
Yeah, no, it's a massive challenge. And I pointed it out to say that there's a lot of new opportunities available in the space, like the area for research, you know, it keeps unfolding to us as we study human milk. But yeah, one strategy is to study a wide variety of milks and digestive products and compare the ability of those milks to exert some sort of function, whatever function you're studying, antibacterial, immune modulation, etc. And complete statistical correlations between the abundances of certain peptide species in those samples with the level of activity of that sample. So having a large set of samples can really help that. And then synthesizing, testing specific peptides that are good candidates for, you know, causing those observations. That's one way of doing it. Other ways are, you know, like focusing on things that are most abundant, things that seem to be most consistent. For example, like peptides that can persist throughout the digestive system. That's a good sort of target to follow up on. So like we've seen some things that show up in the milk, in the stomach, in the intestine, and in the stool. So that can be a really good target. But yeah, it is a big challenge.
Yes, and if you can hear me now?
Yes.
Yes. I guess also on this variability, of course, we need to remember that nature is very variable. And sometimes it's for a good reason. I guess it's always for a good reason, but the reason can be that it doesn't really matter. So sometimes there's allowance for a big variation, and sometimes the variation is critical. You just think about the variation between antibodies that is so functional. So I guess we need science to investigate how important the variation is. Is the average, is that our best choice, or do we actually need a battery of variant structures to achieve the functionality? So
that's a very interesting point. So should we worry about variability, or is this just the robustness of the system, so it doesn't matter too much what we are exactly feeding because the baby can deal with the variability anyway?
Or do you capture the wide variability as much as you can to have a wider net? And then how important is circadian rhythm and the timing and the change, the transition of milk to meet the functions of the growing infant, right? So you have early milk and or colostrum-like material, transitional, is that viable for companies to create such different phenotypically focused products?
But again, I think it's so important to understand how important it is, because there's no way we could capture the complexity and variability of nature and mothers and children and settings and timing and so on. That cannot be reflected in an infant formula. I mean, how many stages should you have? One per day or what? So of course it needs to be simplified. And here I think what is important is to still focus on the gap we see between in the breastfed and infant formula fed children, and we need to work to make that gap as small as possible. And I don't think we should strive to fully mimic nature. I think that's too tough.
Thanks for that. Then there was another question just popping in, which I think links to earlier questions to you, David. It's about computer modeling of digestion of protein profiles. So you can also try to predict which peptides would be formed upon digestion. And could you use something like that to predict also matrix effects or other things that are happening upon consumption?
Yeah, that's a really good question. I mean, at this stage, there doesn't exist anything like that that performs effectively to try to mimic what occurs in an infant or an adult. We have models of cleavage specificities of various enzymes, but the modeling all those different interactions is extremely difficult. But if there's any computer scientists out there in the audience who want to work on that challenge, please let me know. I'd be glad to work with you.
Thanks, David.
I have a question for Daniel about where, and this is actually from Vanessa Castagna, about where is the line between, functions that are food and then functions that are more therapeutics, like antimicrobial functions, for example. Some peptides have antimicrobial functions, and where do regulatory agencies say, okay, you're in the therapeutics section, so you're going to go in a different, this is not food.
In the theory, when you are in the food area, you cannot claim that you treat disease. When you say I treat disease, it is pharmacology, it is drugs. For the food, the limit is that you can only show that you reduce a marker as risk factor of disease. It's preventive. You reduce, even when you show that you reduce the risk of the disease, you can be accused to go to drugs. So you only have to show that you reduce a risk factor of the disease. For instance, the classical example is cholesterol. If you reduce the risk of cardiovascular disease, you are a drug. If you reduce the level of cholesterol, we know that cholesterol is a marker, is a risk factor of future cardiovascular disease, then you are in the food.
Okay, so antimicrobial peptides can target let's say some gram-negative bacteria that may cause disease. Is that food?
The disease is not yet here. So you can show, okay, it's a food. It reduces a microbe that is responsible to induce a disease after.
Okay. Sounds simple. Now all the infant formulas can do.
The theory. After in practice, very often it's much more difficult. I agree, because you have to experiment in population, and in this population, even you have to choose people which are at risk. Because if there is absolutely no risk, you are sure you will not demonstrate anything. The control will be the same that the complemented group and so on. So you have to find people which could have a disease in the future or not so far. But it is the same problem with diabetes. Very often when you want to target diabetes, for instance, you can experiment on pre-diabetic subjects, but you say they are not ill. They are only pre-diabetic. They are at risk of diabetes, but they don't have already the diabetes.
So I do want to talk about sort of the elephant in the room that's quite timely in the United States, is we have an infant formula shortage crisis. And as you know, we have 3 huge infant formula companies in the US that control the market. And Daniel, I was hoping you could maybe talk to some of the smaller infant formula companies out there that are trying to sort of have, provide products to parents, to sort of increase that availability. What suggestion do you have for smaller companies that want to be competitive with some of the larger giants to actually provide a commercialized product for people?
But the problem depends on their formula. Is their formula exactly in agreement with the recommended composition of infant formula? I think they can put it on the market directly. They just have to show that their infant formula is not different than the other formula on the market. The problem is when there is some new formulation, new components in the formula, then both in US and in Europe, they have to apply to the regulatory authority to be allowed to put this on the market. Now, because we know that the infant formula, the regulation is very strict. You have a close regulation. You have a reference composition of the infant formula. If you are in this reference, you can put the product on the market using the ingredients which are allowed by the regulation. We know that milk protein are low, soy protein are low. Now, I think rice protein hydrolysate are low, and so on. So, if you use components which are authorized, it's not a problem. They just can make the formula and put on the market.
No, you still have to do a PER study and a growth study in a 90-day notice, though. Like you still have to, even if you're using components that are already GRAS, you still, if you're formulating a product, you still have to go through all of those steps, which for new companies is long and expensive and doesn't allow us to address the current challenges. Like you can't just
for some safety, probably
Sorry?
not. The main problem is then there is a formula that is not equivalent. For instance, to demonstrate for a formula to be accepted, you only have to show that the Z-score are the same that another formula already on the market.
Yeah, but you have to, as a small company, you still have to run that infant clinical study, right? And compared to the gold standard formula, like, which is typically done. And so, there's several studies right now happening from, new startups in the space where there isn't anything like particularly interesting about the products that they're putting on the market, or like there isn't necessarily new ingredients being used, but they're still running these clinicals. So it's just like a big cost and hurdle to the small startups. At least that's what we've seen.
If they make a formula that is exactly the copy of the traditional formula, I think they don't need a clinical study. They need all the safety aspect. After the factory has to be agreed for safety and so on. There is a lot of rules when you produce infant formula, but not for the formulation.
Interesting. I didn't know that. Like, we've seen people do otherwise. So maybe that needs to be shared more widely with people.
You're muted.
Sorry. Thanks everyone for discussing in this panel session. Also, thanks to the audience for providing the questions that inspired this panel discussion. But now I would like to give the word back to Jennifer for some closing remarks at the end of this workshop. So Jennifer, please go ahead.
Thank you, Casper, and thank you everyone for being here. And on behalf of the IMGC Workshop Committee, we would like to thank each speaker for taking so much time out of their busy schedules to meet consistently, to create their presentations, to engage in this riveting Q&A, and for sharing their invaluable insights. So thank you all. Dr. Tomei shared his decades of experience and insight on navigating regulatory to deliver safe, isolated ingredients to the market. Professor David Dallas highlighted methodological details and analytics that are critical for discovering biological functions of milk peptides and proteins. Laura Katz shared Helena's framework for evaluating the safety of recombinant proteins, which is proving more and more to become a reality for food industry as it has for biotherapeutics. Roland van Eerten shared Friesland Campina's trials and tribulations and successes in bringing lactoferrin as a product isolated from bovine milk to the market. And finally, we end the webinar with Anne Quisgaard and Dr. Peter Weiss, who shared a detailed history, fascinating one, of Arla Foods ingredients, experience and successes in scaling up, analyzing, and navigating regulatory for commercialization of osteopontin. So I would like to also thank the members of the IMGC Workshop Committee, which is a subcommittee of the Scientific Advisory Committee. So each workshop committee member who's circled here has spent several hours meeting and sharing feedback for designing a productive webinar. And of course, I would like to thank our consortium and gold level sponsors, because without them, we could not publish Splash, we could not host the IMGC annual symposium, support the awards program, or pay for this Zoom meeting. So thank you for your support. And then finally, I want to thank all of you, our base, for advancing the field with your passion, dedication, and scientific inquiry in mechanistic discoveries and their translation for human health. There are several ways you can get involved with IMGC. So first, you can subscribe to Splash Milk Science Update. This is our bi-monthly publication. You'll receive a trove of articles ranging from topics such as lactation, milk research, and dairy science. And you can just scan the QR code here on the left-hand side of the screen, and then you can receive the bi-monthly articles. And please share with your network and social media, with your family, friends, and peers. And you can also get involved by completing that 5-minute survey I keep talking about. And this is because it will only improve following, future webinars that we host every year in the spring. You can access the survey by scanning the QR code as well. It's in the middle of the screen. Another way you can get involved is to join the 2022 Webinars Authorship Committee. That's right. So this webinar is we're not ending with just a webinar and a Q&A session. We believe that the goal of the workshop is to bring science to practice. And so the next steps for this year's workshop is to draft a preview article for publication about future directions that we envision for analytics, scaling up, and navigating regulatory for milk peptides and proteins. So if you'd like to join this authorship committee and co-author this preview paper, please submit your contact information in the survey. That's right, that 5-minute survey. Finally, don't forget to register for the 19th annual 2.5-day hybrid symposium at UC Davis this fall, and the details are available on our website. Thank you everyone very much for your participation, and have a wonderful day. Bye everyone.
Thanks everyone for discussing in this panel session. Also thanks to the audience for providing the questions that inspired this panel discussion. But now I would like to give the word back to Jennifer for some closing remarks at the end of this workshop. So Jennifer, please go ahead.
Thank you, Casper, and thank you everyone for being here. And on behalf of the IMGC Workshop Committee, we would like to thank each speaker for taking so much time out of their busy schedules to meet consistently, to create their presentations, to engage in this riveting Q&A, and for sharing their invaluable insights. So thank you all. Dr. Tomei shared his decades of experience and insight on navigating regulatory to deliver safe, isolated ingredients to the market. Professor David Dallas highlighted methodological details and analytics that are critical for discovering biological functions of milk peptides and proteins. Laura Katz shared Helena's framework for evaluating the safety of recombinant proteins, which is proving more and more to become a reality for food industry as it has for biotherapeutics. Roland van Eerten shared Friesland Campina's trials and tribulations and successes in bringing lactoferrin as a product isolated from bovine milk to the market. And finally, we end the webinar with Anne Quisgaard and Dr. Peter Weiss, who shared a detailed history, fascinating one, of Arla Foods ingredients, experience and successes in scaling up, analyzing, and navigating regulatory for commercialization of osteopontin. So I would like to also thank the members of the IMGC Workshop Committee, which is a subcommittee of the Scientific Advisory Committee. So each workshop committee member who's circled here has spent several hours meeting and sharing feedback for designing a productive webinar. And of course, I would like to thank our consortium and gold level sponsors, because without them, we could not publish Splash, we could not host the IMGC annual symposium, support the awards program, or pay for this Zoom meeting. So thank you for your support. And then finally, I want to thank all of you, our base, for advancing the field with your passion, dedication, and scientific inquiry in mechanistic discoveries and their translation for human health. There are several ways you can get involved with IMGC. So first, you can subscribe to Splash Milk Science Update. This is our bi-monthly publication. You'll receive a trove of articles ranging from topics such as lactation, milk research, and dairy science. And you can just scan the QR code here on the left-hand side of the screen, and then you can receive the bi-monthly articles. And please share with your network and social media, with your family, friends, and peers. And you can also get involved by completing that 5-minute survey I keep talking about. And this is because it will only improve following, future webinars that we host every year in the spring. You can access the survey by scanning the QR code as well. It's in the middle of the screen. Another way you can get involved is to join the 2022 Webinars Authorship Committee. That's right. So this webinar is we're not ending with just a webinar and a Q&A session. We believe that the goal of the workshop is to bring science to practice. And so the next steps for this year's workshop is to draft a preview article for publication about future directions that we envision for analytics, scaling up, and navigating regulatory for milk peptides and proteins. So if you'd like to join this authorship committee and co-author this preview paper, please submit your contact information in the survey. That's right, that 5-minute survey. Finally, don't forget to register for the 19th annual 2.5-day hybrid symposium at UC Davis this fall, and the details are available on our website. Thank you everyone very much for your participation, and have a wonderful day. Bye everyone.